Good morning, students. In the last class, I had told you about the lipid component of the membrane. Today, I will tell you about protein and carbohydrate. Proteins are the main component of cell membrane. They are extracted by dissolving the membrane in sodium dodecyl sulfate, which is an ionic detergent, and then separated by polyacrylamide gel electrophoresis. Some of the functions of membrane proteins are they impart strength to the membrane. They play a structural role in stabilizing and shaping the cell membrane. They act as transporting substance, carrier protein or transport channels. I will tell you about the transporting substance in the section Transport across membrane. They serve as enzymes, as an antigen and as receptor molecules. There are two types of proteins, transmembrane protein and peripheral protein. This classification is based on the degree of association of proteins with the membrane. Transmembrane proteins constitute 70% of the total membrane protein, while remaining 30% is the peripheral protein. Transmembrane protein is comprised of hydrophobic amino acids, while peripheral proteins are comprised of hydrophilic amino acids. Transmembrane proteins are found in association with lipids since they are comprised of hydrophobic amino acids, while peripheral proteins are free from lipids. Transmembrane proteins are insoluble in aqueous solution, while peripheral proteins are soluble. Transmembrane proteins extend throughout the lipid layer, projecting out on either side, both on the extracellular surface as well as towards the cytoplasm. Peripheral proteins are present outside the lipid bilayer. Transmembrane proteins can be extracted by treating the membrane with hydrophobic bond breaking agents, while mild treatment with aqueous solution of high ionic strength or alkaline pH is sufficient to extract peripheral or extrinsic protein. Transmembrane protein has got across the membrane function, while peripheral protein, as the name is suggesting, has got peripheral function. Some of the examples of transmembrane proteins are blood group antigens, hormones and drug receptors, while peripheral proteins, spectrin, cytochrome C, etc. Here's the diagram showing the arrangement of integral and peripheral membrane proteins in association with the biomolecular layer of phospholipid. The diagram clearly shows the arrangement of integral protein and peripheral protein. Types of transmembrane proteins. They are of following types: monotropic, bitropic, polytropic, porine or barrel protein. Monotropic transmembrane protein. They are rare. They remain embedded in the lipid bilayer, exposed at only one membrane surface. Example is cytochrome B5. Bitropic or single pass transmembrane protein, comprised of single transmembrane segment exposed on both sides of the membrane. They help in transmitting message from the cell surface into the cytoplasm. Example is glycophorine. Polytropic or multi-pass transmembrane protein, comprised of more than one transmembrane segments exposed on both sides of the membrane, forms channels for the passage of ions and solutes across the membrane. Example, proteins forming ion or solute channels, signal receptors in endoplasmic reticulum membrane. Porine or barrel proteins, another type of transmembrane protein. Proteins in the form of beta sheet or beta barrel form pore in the lipid bilayer. This allows hydrophilic solutes to cross the membrane. Example is aquaporine. The diagram shows different types or forms of transmembrane proteins. Now, interaction between transmembrane protein and lipids. There are two types of interaction: ionic interaction and hydrophobic interaction. Ionic interaction is between hydrophilic side chains of protein and polar head of lipid, while hydrophobic interaction is between hydrophobic side chains of proteins and non-polar tails of lipid. Now come to types of peripheral protein. They are of three types. Covalently linked cytosolic extrinsic protein located on the cytosolic surface, covalently linked to fatty acid side chain. Then covalently linked non-cytosolic extrinsic protein located on the extracellular surface of the lipid bilayer attached covalently. Third type is non-covalently linked protein found on both the surfaces, non-covalently linked to transmembrane proteins. The diagram shows the asymmetrical distribution of membrane proteins. Most of the membrane proteins are located on the cytosolic surface. The diagram clearly shows this. Now the lipid anchored proteins. They are another type of protein linked to lipids. They are located outside the lipid bilayer, covalently linked to a lipid. They fall under two categories. They may be present on the extracellular phase as well as towards the cytoplasm. So depending upon this, two categories are proteins on the extracellular phase and proteins on the cytoplasmic phase. Proteins on the extracellular phase are bound to plasma membrane by a short oligosaccharide linked to a molecule of glycophosphatidyl'inositol that is embedded in the outer leaflet of the lipid bilayer. Proteins on the cytoplasmic phase of plasma membrane is linked to a long hydrocarbon chain embedded in the inner leaflet of the lipid bilayer. Now come to enzymes and carbohydrate. There are about 30 types of enzymes. Since enzymes are proteins, so as proteins are asymmetrically distributed, so enzymes are also asymmetrically distributed more on the cytoplasmic phase of the membrane. Some important enzymes, especially of RBC, are acetylcholinesterase, protein kinase, adenylate cyclase, acid phosphatase. Carbohydrate constitutes about 1% of the total. They are covalently linked to ectoprotein, proteins located towards the extracellular surface. Carbohydrates are also linked to the polar heads of the lipids at the extracellular surface. They are present as short branched or unbranched chains of sugars. Examples of carbohydrate are D-galactose, D-mannose, L-fucose, N-acetyl-neuraminic acid, etc. Little more about carbohydrate, glycoprotein and glycolipid. They are carbohydrates linked to protein and lipids respectively. Glycoproteins help in recognizing own cell type. This has got significance in blood grouping or in rejection of transplant. Glycoproteins act as cell receptors. Glycolipids. They function as gate for the entrance of many pathogens and as receptor in normal cell function. Both glycoprotein and glycolipids form cell coat or glycocalyx. Same as lipids and proteins, carbohydrates are also asymmetrically distributed. Carbohydrates are found only on the external surface of the membrane. So this is about the different components of the membrane. Thank you.